Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into med school and other professional programs. Welcome to another enlightening episode of MCAT Bites. Today, we dive into a topic that sits at the fascinating crossroads of genetics and epigenetics, genomic imprinting. This is one of nature's curious genetic playbooks, influencing how we inherit certain genes from our parents. Let's begin to peel back the layers of what I found to be a really confusing topic when I was studying for the MCAT. So first, let's kind of provide some background information. Let's set the genetic stage. Every individual inherits two copies of their genes, one from the mom and one from the father. Normally, both of these copies are active and contribute to the individual's phenotype. But imagine a scenario where only one of these copies is active and the other remains silent, regardless of its sequence. That's genomic imprinting in a nutshell. But we need more than a nutshell for the MCAT, so we'll dive a bit deeper. Genomic imprinting is an epigenetic phenomenon, right? Because it can't be genomic if we have two different genes. It has to be something higher, epi, above genetic, epigenetic phenomenon, where genes are expressed in a parent of origin specific manner. Think of it like a genetic switch. If the allele from the father is imprinted, it is silenced, and only the allele from the mother is expressed, and vice versa. So why does this happen? What are the origins of imprinting in evolutionary biology? There are a couple of theories, but my favorite is that it's a tug of war between maternal and paternal genes. For instance, paternal genes might favor extracting more resources from the mother during fetal development, while maternal genes might regulate this to preserve resources for future offspring. So what are some of the mechanisms behind the scenes? Because as cool as the idea of a genetic on and off switch is, imprinting involves epigenetic markers during egg and sperm development. During egg and sperm development, certain DNA regions acquire chemical tags, like methylation. This dictates which gene copy gets shut off in the offspring. Importantly, these imprints are erased and reset during the formation of eggs and sperm, ensuring the correct paternal imprints are established in each generation. So as you may be thinking, there are a lot of significances in development. Imprinted genes can play crucial roles in growth, development, and resource allocation. This makes sense when you think of it as the evolutionary battle for resources between maternal and paternal genes. Let's take a look at an example here. Consider a hypothetical gene X. If a maternal copy is imprinted and hence silenced, which copy would be the active in offspring? Well, in this case, mom's going to be off, so dad is going to be on, so we're going to have the paternal copy of gene X active in the offspring. And I don't know about you, but I didn't take the MCAT to just memorize random things. I wanted clinical applications, right? You want to be a doctor, so who cares? Well, errors in imprinting can have huge, dire consequences. Two of the most famous ones are Prader-Willi and Engelmann syndrome. Both of these arise from the deletion of the same gene on chromosome 15. However, the difference lies in which parent's copy is deleted. Prader-Willi results when it's the paternal copy, and Engelmann when it's the maternal copy. Similar disease is Beckwith-Wiedemann syndrome. This is linked to abnormalities in imprinted genes on chromosome 11, leading to overgrowth and increased cancer risk. And I don't want to scare you, don't memorize these diseases, you don't know them for the MCAT. You might once you become an actual doctor, but for now, it's just kind of nice to know that this is an important phenomenon. And it's also important for you to tackle problem 2 here. If a child is born with Engelmann syndrome, which parental copy of the relevant region on chromosome 15 is likely intact? Well, once again, I would think it'd be the paternal copy would be intact, as Engelmann arises from a deletion in the maternal copy. Understanding genomic imprinting isn't just about acing the MCAT, even that's a big part of it, or even just gaining genetic knowledge. It sheds light on complex inheritances and offers insight into certain developmental disorders and underscores the intricate dance between genetics and epigenetics. So, conclusion, in 4 words, our genome is complex. Genomic imprinting serves as a reminder of the intricate and nuanced ways our genes are regulated, influenced by both ancestral battles and molecular markers. As you prepare for your future in medicine, such foundational knowledge forms the bedrock for understanding genetic disorders and patient care. Until our next genetic journey, stay curious and keep studying.